But first of all, I would like to thank Drexel and Chris and all the other staff of De La Salle University Central Instrumentation Facility for hosting me as Balik Scientist. Before I make any of my presentation, I just want to give you a overview of what I am about to present this evening. So there are 3 presentations. They're all focused in terms of analytical chemistry, and then in the background of pharmaceutical science. The methodology is heavily focused on chromatography. Now, for the first presentation, which is the analytical method lifecycle management, this is much more applicable to those who have a bit advanced experience in the pharmaceutical industry. Those are the people who actually submits a dossier to the FDA or interact with the pharmacopoeia. The second one, which is the development of alternative procedure in analytical scientists, is more towards those who are now using the United States Pharmacopoeia monographs and general chapters, but have products that have some kind of a difficulty. So in this presentation, I'll try to lead you into how you can go around and make your methods and procedures successful in terms of utilization in the characterization of your product. The last one, as we did this in reverse, the last one is very much more on the basic. So this would help a lot of those people in the academia also and in industry in terms of how the important parameters in terms of developing and also validating analytical procedures and also transferring it from one site to another. So without much further ado, let me start with my presentation. Okay, I think I have my presentation ready. So the first presentation is about the analytical method lifecycle management. And this is focused on a new proposed general chapter in USP, United States Pharmacopoeia, General Chapter 1220. But I will be providing you with compendial perspective. So, but before I make any of my presentation, I would like to make a disclaimer that the presentation that I'm about to give you are my views and should not be construed as views or policies of the United States Pharmacopoeia, US FDA, or International Conference on Harmonization. In terms of the outline of my presentation, I will first discuss some of the references because this new proposed general chapter in USP came in as a stimuli article. Also, afterwards, before I actually go into the details describing you this general chapter, I will provide you with some background in terms of the current analytical method compendial management. It's a long title, but these are the processes and challenges of implementing new method in a laboratory. I will then move on and describe to you the proposed USP General Chapter 1220 Analytical Procedure Lifecycle, and discuss with you the potential changes in terms of compendial standards. Finally, at the end, I will try to summarize and provide you with some of the advantages and possible changes. So as an introduction, let me just ground everyone in terms of USP, United States Pharmacopoeia. So only in United States is where the FDA is separate from the Pharmacopoeia, because the US Pharmacopoeia was established 200 years ago, way ahead of the US FDA. But the US FDA uses the standards that the USP creates to ensure the quality of drug products in United States. And I believe many other countries, including the Philippines, uses USP as part of their standard setting for drug products in the Philippines. So in the book of USP, there are three important things that you need to know. One would be the general notices. So the general notices in USP contains requirements that are applicable throughout the book of USP, but it is superseded, meaning a more detailed instruction is given through what is called the general chapter or monograph. So in the general chapter in USP, these are the requirements that are applicable to many of the monographs. When we say monographs, at the end, I'll describe to you what it is, but the general chapter supersedes the general notices. And general chapters in USP are referenced in monographs. If they are numbered below 1,000, they are term enforceable chapter. If they are numbered above 1,000, those are general chapters that are informational chapters. General chapters can be procedural, which means they tell you a procedure, a test method, but they can be a guidance general chapter. And finally, we go down to the one where it supersedes both the general notices and the general chapter, which is the monograph. So the monograph contains the requirements that are specific to the article. When I say article, it's referring to the drug substance or active pharmaceutical ingredients, API, or the drug product, or what some term as finished dosage forms. So the monograph contains all the requirements and it supersedes the general notices and general chapter requirements. In terms of references, where before the new proposed General Chapter 1220, it's called the Analytical Procedure Life Cycle, was published in PF, Pharmacopoeial Forum 43.1. There were publications that were published before it and stimuli articles that were published after it. So these publications that were published before are stimuli articles that have important terminologies. If you want to find out more about the new General Chapter 1220, you can read more some of the background information that were published before as listed in this slide. So it contains method development procedure, performance qualification, performance verification, and then you have the target measurement uncertainty, the analytical target profile, and the analytical control strategy. Afterwards, there were also stimuli articles that were published after the publication of the PF 43.1, which is proceedings of the workshop and also the measurement uncertainty for the pharmaceutical industry. So I apologize. Some of the jargons may be not familiar to many of you because I have the audience have a varied experiences, but you can always send your questions at later time, okay, after my presentation, and we can answer some of those. The goals of the analytical lifecycle management is that the different general chapter guidances that USP provides in terms of the validation, verification, and transfer of analytical procedures were written at different times, and they were done separately from one another. So they are not very much well integrated, and I will show you why. So therefore, there's a new way of working that has been proposed, which is this General Chapter 1220, that is more effective and efficient through an interrelated and a continuous process. Also, the increasing the knowledge of the procedure that contributes to the different sources of variability for a method will result in fewer failures during qualification or verification. And then the design of more robust procedure or method will also result in fewer failures such as system suitability. And finally, if there is an increase in the facilitation of continuous improvement and change control, will provide a better way of working in terms of method development and validation in a laboratory. Utilization of new analytical method versus traditional HPLC. So I've been in the pharmaceutical industry or in as an analytical chemist in the United States for more than 30 years. And it's been HPLC, high performance liquid chromatography, that has been used really as a workhorse. Although there are other new sophisticated instrumentations available, but industry wise, in a practical sense, it's always the traditional HPLC. So I the production of pharmaceutical may be changing in the future, such as the continuous manufacturing. That's a terminology. So a new analytical method would be something that is going to be useful. Now, the analytical life cycle management may result in the additional effort in terms of understanding the guidance and regulations. So the guidances are more on the regulatory and regulations, adapting new ways of working. So in a setting, in an industry lab, for example, or even in regulatory lab, always a method goes through a method development and validation. It may change based on this new proposed general chapter. And then finally, acquiring the knowledge and skills that is required to comply. With this new proposed general chapter, there may be requiring people to know more about statistics, design of experiments, quality by design, and risk assessment of different situations. So the approach for this new proposed general chapter 1220 is holistic, meaning it is covering all the different activities from method development to validation, routine use, change control, until the method is retired. And this enhanced approach improves the method understanding and performance that is leading to, first, fewer out of specification results. You guys in the industry knows what is this out of specification results. It facilitates method transfer. So you're moving one method, one procedure from one site to another. And also the potential to lessen the regulatory burden. So this is something that is very much of interest in the United States, because the FDA really goes after the industry in terms of how they manage their product. So before I describe to you the general chapter 1220, what I wanted to do is to provide you with some background in terms of current implementation of new analytical method in a laboratory. So if you're, for example, an industry, a pharmaceutical industry, you are using different methods to analyze your sample. Your methodologies can either be coming from the USP method, which is the book. It's termed compendia. But at times, there are methods that are not in USP. So those are termed non-compendial or alternative methods. Those are the internal methods from your company. However, this will have to adapt into the new proposed General Chapter 1220, starting with the establishment of what is called the Analytical Target Profile, ATP, and Target Measurement Uncertainty, TMU. These two parameters is consistently the standard in the 3 stages of the procedure life cycle, starting with stage 1, which is procedure design and development, having 5 different elements. Stage 2, which is procedure performance qualification, having 3 elements. And then finally, stage 3, which is termed as the continued procedure performance verification. Again, it has 3 elements. These 3 stages, stage 1, 2, and 3, are all interconnected with each other, so as to provide a continuous process of improvement. So it's not one way, but it could cycle back and forth. So we go to the current implementation of new analytical method in a laboratory, either by using a USP method or an alternative method. This involves actually examining or having an understanding, for most of you who are not familiar in the pharmaceutical industry, goes from the drug development, starting from drug development, going into the preclinical testing, and then to what is called the investigational new drug, and then FDA reviews them And it goes to the 3 different clinical phases, phase 1, 2, and 3, until it becomes a new drug application. It's continued on in terms of the review from the FDA until the analytical method is finalized before the company gets approval for marketing. But once the drug is in the market, there may be some changes in terms of the manufacturing after post-approval changes. Now, United States Pharmacopoeia is focused when the drug is in the market. That is, we set the standards for those pharmaceutical products that are in the market. So industry-wise, the pharmaceutical industry is focused in terms of regulatory requirements, starting from the drug development all the way until the drug is approved. So I wanted to make that clarification between the regulatory FDA and the compendial USP. So just as a summary, a method typically goes from the development stage to validation. So the validation guidelines based on USP is General Chapter 1225. So this procedure or methods are routinely used in a laboratory. Either there's a need to do a verification of the product being produced, or there is a need to transfer the analytical procedure, for example, from a research lab to a manufacturing site. The USP General Chapter Guidance created at different times to address the needs of the customers, but they are not well coherent, as I mentioned earlier. So those 3 general chapters, starting with the USP General Chapter 1225, describing the validation of compendial procedures, is heavily focused on chromatography, I mentioned earlier, and was created before the ICH-Q2, which is a description of how to do method development and validation. ICH, by the way, is International Conference on Harmonization, and it is more of the international standard for drug products and drug substances. So it covers the different countries. So that's more of United Nations, but that covers the major USP, British Pharmacopoeia, and also the Japanese Pharmacopoeia. Now, USP General Chapter 1226, which is the verification of compendial procedures, were created to support the needs of the stakeholders, and the General Chapter 1224, which is the transfer of analytical procedure, is the one that has been recently added as a general chapter, because for many years, organization just move one method or procedure from one site to another without much formal process. So what is the process of implementing new method in a laboratory, either compendial or alternate or harmonized? So this slide, this is next slide, will describe to you the process. So when you are implementing, for example, a procedure coming from the USP book, which is termed compendial, and you're analyzing your product, you must do verification. But this is only the first time, one time. When you're doing verification, you need to do documentation and determine if the method is suitable to analyze your sample. If it is suitable, then you can do implementation. But if it is not suitable, and because you are using, for example, an HPLC method, you can use what is called the USP General Chapter 621, or the System Suitability Adjustment. So you can make those adjustments and determine if the method is appropriate to analyze your sample. If it is suitable, then you can do the implementation. And if not, then you'll have to contact United States Pharmacopoeia for possible alternate method or procedure, or even the creation of what is called a flexible monograph. Now, if you are implementing an alternative method, which is termed internal to your company, it is important that these methods and procedure must be validated so as to be able to do any transfer to one side to another. If they are not validated, then you need to validate them based on the General Chapter 1225 method validation. I will discuss this at the last presentation. And in terms of transferring, there are 4 different ways of transferring an analytical procedure, but they are again based on whether the method is validated or not. If the method is validated, for example, and everything is the same, meaning the instrumentation, the staff, and the procedure doesn't have much issues, then there's what is called the transfer waiver. So you can just transfer it without much documentation or experimentation. If not, then you can analyze a common sample between a transferring lab and a receiving lab, and this is termed as the comparative testing. And if not, if you still have issues, you have to do what is called the revalidation, meaning the receiving lab has to do or some or most of the validation processes. If the method to be transferred is not validated and the transferring lab is available to do some of the validation, then there's the 4th option called the co-validation. So this is a collaboration between the transferring and the receiving lab. So these are the 4 options in terms of a formal movement of a method or procedure from one site to another. So what are the ways to deal when you come up with issues implementing a new method or procedure? So when I was working as a scientist at United States Pharmacopoeia, I do get some calls from the industry in the Philippines asking questions about some of the difficulties that they have. Just to clarify, if you are having difficulty and you're using HPLC and you're using a USP monograph, you can use what is called the USP General Chapter 621. But this, again, is only applicable to USP monograph. If not, if you're using an alternative or harmonized method, one thing you could do is to make the adjustments. But remember that these methods that are not reflected in USP monographs are subject to regulatory review. So FDA can ask you questions about the validity of your method. So these are not reflected in USP monograph. The 3rd way would be, if you want, you can contact USP so that your modified alternative procedure can be added to the USP monograph, simply because you have a little bit of a different product compared to the other products. But this would require a lot of collaboration with USP staff. So these are the ways to implement new methods if you're having issues. Now, this slide is a summary of what is called the System Suitability Adjustment Guidelines for HPLC. Now, the first column lists all the variables in HPLC. The second column are the allowable range of adjustments. So you can make adjustment changes. If you are using a USP monograph, you can apply these allowable adjustments. And the nice thing about it is you do not have to do any validation. You can apply, and then if it works, then you're fine. Keep in mind that these allowable adjustments are limited to whether your system is isocratic or gradient. Some of them allows either isocratic or gradient, but some of them are only limited to isocratic. So when we talk about isocratic, meaning it's one mobile phase in HPLC. If it's gradient, the mobile phase is changing. So anyway, this is just a summary of what is available for the USP General Chapter 621. Also, keep in mind that, hold on. I guess I cannot go back. In the previous slide, keep in mind that, thank you. Keep in mind, if you are using other pharmacopoeia, for example, like the Japanese pharmacopoeia or British pharmacopoeia, the adjustments for system suitability are different from USP. Japan has their own adjustment guidelines, and British pharmacopoeia have their own adjustment guideline. Thank you. So let me go to the next set of slides, which is discussing the analytical procedure life cycle. As I have described to you, the implementation of a compendial or non-compendial methods in the laboratory, I mentioned earlier that this have to be adapted into the new proposed life cycle, starting with the analytical target profile, ATP, and also target measurement uncertainty, going to the three different stages, stage 1, 2, and 3, which are all interconnected to each other for to provide a continuous process of improvement. Let's talk about the analytical target profile. Analytical target profile states the required quality of the results produced by the analytical procedure in terms of acceptable error in the measurement. Also, it states that the allowable target measurement uncertainty associated with the reportable value. And then because of the ATP describes the quality attributes of the reportable value, it is applied during the procedure life cycle and connects all the three stages. I mentioned that stage 1, 2, and 3. And then the ATP criteria are derived from external requirements and not only from the performance of the analytical procedure. If you are familiar with method validation, you understand the use of what is called the analytical performance characteristics, the accuracy, linearity, limit of detection, so on. But those are what is termed as internal requirements. The ATP is derived also considering the external requirements. The external requirements, as when we discuss again, verification, those are focused on the method and the sample. Those two things, method and the sample. The acceptable level of risk in making an incorrect decision is considered when establishing ATP, which means you need to have some level of understanding in terms of statistics. And then the reportable value may be the mean of multiple analytical results. So a replication strategy, number of trials have to be defined in the analytical target profile. So when you're establishing the ATP analytical target profile, it's important to consider sample to be tested, matrix in which the analyte will be present, the allowable error for the measurement as assessed through the accuracy and precision. These are the ones that make up the target measurement uncertainty. Also, the allowable risk criteria that are not being met. And then finally, assurance that the measurement uncertainty and risk criteria are met. So those are the things about the ATP. We go into the target measurement uncertainty terminology. TMU is the maximum uncertainty that can be associated with a reportable results while still remaining fit for purpose. So TMU is a comprehensive term versus the traditional term for just the precision, which is representing random errors, and the bias, which is more on the accuracy. So TMU can be considered to represent all the associated factors that affects the ATP. So the two main elements in target measurement uncertainty would be the bias or accuracy and precision. So in this next slide is a consolidation of attributes contributing to the TMU in terms of the accuracy and precision. So list of accuracy is listed here. You have the selectivity, linearity, extraction, efficiency, and everything else. So also in terms of precision, those that needs to be considered would be sample preparation, weighing, instrument, integration, and so on and so forth. So you notice that these elements or these listed factors for both accuracy and precision are not limited to only the internal to the method, but it is also external to the method, which covers both, again, the methodology and also the sample. let me discuss with you briefly the three different stages. So in the first stage of the proposed general chapter, which is the procedure design and development, 5 elements, where the first one would be knowledge gathering. This would be the stage where you're trying to determine what kind of methodology, what kind of procedure am I going to use? Do I use a HPLC or near IR or Raman? The next step would be to find out the risk assessment, because after knowing all the relevant information in the first element, the knowledge gathering, then you determine what are the risks in terms of the variables that will impact my reportable values. You may be using what is called an Ishikawa diagram or the design of experiment tools. If you're not familiar, you can learn those things. The analytical control strategy would be the third thing in the stage one, which is you are going to be establishing a planned set of controls derived from the requirements for fitness for purpose, meaning what are your control strategy so that you can maintain the ATP, analytical target profile, and the target measurement uncertainty. Fourth one would be your replication strategy. Again, this is trying to cover the statistical analysis of the samples. The last element in the stage one would be the knowledge management. This would include acquiring, analyzing, storing, and disseminating information related to the activities involving the development, technology, transfer, qualification, and change management. So this is something important also. After you complete the stage one, it is good to evaluate the data that you obtain in terms of the absence of significant bias so that your precision is at the appropriate level. And then you, before starting the qualification, which is stage two, you can perform a compendial procedure from other pharmacopoeias such as Japanese pharmacopoeia, EP, BP, other than USP. Also, you can perform what is called a quality risk management. And then the third one would be to verify control strategy of the procedure so as to ensure that the analytical target profile are met. So when you start stage two, which is the procedure performance qualification, it has 3 elements. The first one is to establish a protocol. What are those protocols? Protocols are those that contain the ATP, the method specific performance, the description of the procedure, description of the experiment, test samples, all the statistical approach to be used for data analysis. This is followed by the qualification, study design, and execution. This step actually confirms that the procedure generates the reportable value that meets the ATP criteria and that it remains appropriate for the testing of the product in the environment that it will be used. So remember, the environment is actually the method and the sample. And then finally, the third element in stage two is to write a final report. So after completing the stage two, one would need to refine and update the analytical control strategy. Because, for example, this would include, let's say, you wanted to have additional control so as to reduce the sources of your variability in a routine operation, and you can modify the replication strategy or replication level. Say you can increase the number of injections, you can increase the number of sample preparations, and these are all based on the uncertainty of the reportable values. When you start the stage three, it has its term as the continued procedure performance verification. It has 3 elements. The first one would be the routine monitoring and trend analysis. So during this time, it's important to determine sorry, was I not, was I muted, Chris? Chris?
Okay, can go on po.
Can I continue?
Yes po, yes po.
Okay. Okay. So continuous improvement would be the second element. So as after determining the different out of specification or out of trend investigation, um, the continuous improvement can ensure that the analytical procedure remains in control, and then a change control is needed at the end so as to ensure that the level of activities confirm the level of change in producing fit for purpose in terms of risk associated with change and knowledge available about the procedure and the effectiveness of the control system. In terms of the potential changes in compendial standards, I'll provide you with an example of the current assay procedure. I'll also provide you with how additional control ranges can be included in the monograph, and finally, the addition of analytical target profile. So this is an example of a current assay procedure from USP, where I put together the sample and standard solution and the chromatographic system, the mobile phase, system suitability, analysis, and acceptance criteria. So addition of control range would be good to add in monograph. For example, in this example that I gave you, the sample and standard solution can have a table that contains parameter. For example, it can have the solvent composition, mixing time, and heat on steam bath, and it would have a target value with a low range and upper range. Similarly, in the chromatographic system, it can have several parameters that would have a target value with a low range and an upper range. Addition of analytical target profile is described in this next slide, where the results are shown, the 4 possible outcomes of reportable results, and the expanded uncertainty as described by the Gaussian curve are compared to the specification. So if your specification is the upper limit, there are 4 possible results, where the first one would be a result that is above the limit. The second is the results is above the limit, but the limit is within the expanded uncertainty. So you can see with this intersection between the Gaussian curve and the upper limit. A third result could be the results is below, but there is also the possibility of the limit is within the expanded uncertainty. And the fourth one is the result is below the limit. So the question is really how much overlap is acceptable between these two situations. So an example of assay procedure with analytical target profile is shown here. The analytical method is capable of quantifying metoprolol target in metoprolol tartrate tablets from 90% to 110% of the true value with an accuracy and precision such that the results reside within not more than 3% with 95% probability. Also, to include in the analytical target profile, this ATP for the content of metoprolol tartrate in metoprolol tartrate tablet is included in this monograph for the convenience of users for which the following procedure conforms. And finally, if you have an alternate procedure, it would say that the alternative procedure should be demonstrated to conform with these requirements. So in summary, this new proposed general chapter is focused on the future trend of the analytical procedure development and quantitation and continued monitoring. So advantages of this approach, again, is to have an improved understanding of the procedure and control the sources of variability. It will produce a more robust procedure. There will be a reduction of overall resources that is required to revise the procedure, and also identification of any adverse trend and continued improvements. Possible changes in terms of USP or compendial requirements would be the information regarding the method development may be required when submitting your dossier to USP. There may be an increase in the use of non-conventional procedures. These are what is termed as the multivariate near-IR or Raman spectroscopy. There may be a reduction in terms of the frequency of monograph revision, enhance the use of quality by design, QBD approach, and finally, minimize the challenges in the compendial procedures for especially the OTC products in the United States. So these are some of the difficulties, and I can discuss this at a later time if you are interested in the challenges for the OTC drug products in the United States. But at this point, I thank you for your attention, and we can discuss some of your questions at the end. Chris?
So now we can now proceed to the second topic of the webinar. Dr. Lionel.
So my next presentation is the development of alternative procedure in analytical sciences. Again, this is focused on the pharmaceutical industry. And again, the disclaimer that I have made earlier that my presentation are my views and should not be construed as views or policies of the USP, FDA, or ICH. In terms of the outline of my presentation, I will briefly discuss the validation, verification, and transfer of analytical procedure, which I will be presenting in the next presentation. Also, again, mention about the process of using a new analytical method or procedure. The one that I will be starting to describe to you in terms of the development of an alternative procedure for test procedures such as those in the identification, assay, and organic impurities. And finally, describe to you alternative procedures for residual solvents and elemental impurities. I chose this because these are the ones that are major in terms of the need in the pharmaceutical industry at the moment. So in terms of the validation, verification, and transfer of analytical procedure, can we go back, Hazel, to the first one? Okay, thank you. So in terms of the validation, USP General Chapter again is validation is an established laboratory studies based on the performance characteristics of the procedure, and it is meant so that it will meet the requirements for the intended analytical application. 1226 is only selecting some of the analytical performance characteristics. It is not a repetition of the validation, but it is intended so that when you do verification of your product, it is trying to ensure that the lab is capable of performing the method reliably and precisely for its intended use. The transfer I mentioned to you earlier, I'm not going to repeat the transfer of analytical procedure again, but this is simply the from a transferring unit to a receiving unit, transferring an analytical method. It is there to ensure that the receiving unit has the procedural knowledge and the ability to perform the transferred analytical procedure as intended. And it is not applicable to compendial procedures or monographs. So I did describe to you the process of implementing new analytical procedure if it is a compendial. And this is shown by this slide. Similarly, implementation of an alternative method for non-compendial or alternative method, as I mentioned earlier, is shown here. And then the ways to deal with issues in terms of implementing can be using the system suitability, using developing the alternative or harmonized method and procedure, which is what I'm going to discuss in this presentation. And then the third one would be what is called the flexible monograph. So in the development of alternative procedure for procedures or methods for identification tests, assay, and organic impurities, you can use the first strategy, which is using the allowed variation in HPLC, or the USP General Chapter 621 in chromatography. Or you can use the development by using robustness study approach. Or you can use both A and B. But keep in mind that when you are developing an alternative procedure, it means that you have an initial knowledge of the procedure based on the monograph. So it's not, you don't have anything. You have something where your starting point would be the USP monograph, except you're having difficulty, so you're trying to modify the method. So in the development of alternative procedure, again, if you're using the General Chapter 621, remember that it is chromatography based. It is a system suitability adjustments, and the user should verify the suitability of the method under the new condition by assessing relevant analytical performance characteristics potentially affected by the change. So again, you can use these allowable adjustments without revalidation. However, if the adjustment is beyond the allowable range, then you need to do some level of revalidation. And finally, the advantage of this using the General Chapter 621 is that your adjustment may be a bit different from what is in the compendial, but it is going to, your alternative method will come up to be very close to what's in the compendial. So that eliminates a lot of your need to do method development, going back and doing a lot of the method validation. So in the system suitability adjustments, you have the list of variables listed here with the adjustment. So again, I mentioned to you earlier that you have to pay attention whether your system is isocratic or gradient, because some of them are only limited to an isocratic system. So I showed you this slide earlier, but this is more on the allowable adjustments, and then I'm highlighting for only those systems that are isocratic and isocratic or gradient. There is a difference between system suitability adjustment between different pharmacopoeias. If you're using the second strategy, which is developing alternative procedure for identification tests, assay, and organic impurities using robustness study approach. So robustness is actually a test procedure or a methodology where you examine the important parameters that affects your method. So for example, if you change the temperature, is it going to be critical? So those are what is termed robustness study. And the next table will show that sample preparation variables can be adjusted or can be revised based on the robustness studies. And these variables may be used as your starting point in the development of an alternative procedure. So in this next table, it'll show you the different factors, what you can do to adjust your procedure. Percentage of organic modifier, for example, you can, and also there's a suggested limit factor level. Sample matrix concentration, you can make this adjustment. pH of the extraction buffer, concentration of the extraction buffer, mixing, equilibrium time. There are many of these factors that is listed in this table. Extraction temperature, dilution ratio, more type of mixing, shaking period, blending time, concentration of the active ingredients, filter pressure, discarding the filtration volume. So, and then finally, the equipment that you use. So you either use a mechanical shaker or blender. So these are all the different factors in terms of how you prepare your sample so that you can come up with an alternative procedure. And you, again, you can combine this with the first strategy, which is the system suitability adjustments. Now, how the next slides will be alternative procedure for developing residual solvents. So in USP, there's a general chapter 467 that describes to you the residual solvent analysis. Again, the La Salle University Central Instrumentation Facility has this instrumentation called the Headspace GC. But in terms of the guidelines on the alternative procedure for residual solvents, it is consistent between ICH, European Pharmacopoeia, and USP that the manufacturer are free to select the most appropriate method to validate uh, in terms of the analytical procedure analyzing for residual solvents. So meaning you do not have to analyze all the residual solvents, but you only need to analyze the residual solvents that are specific to your sample. So in terms of the overview of developing residual solvent procedure, you need to determine what solvents that you're going to be looking for, because there are a lot of solvents and there are three classifications. You need to choose the appropriate testing procedure for those specific solvents. Either you use what is prescribed in the USP General Chapter 467 as written, but you can modify them, or you can come up with your own method of residual solvent analysis. So when you're starting to do the residual solvents and you're the one who produces your own active ingredients or the API or the drug substance, ask the organic chemist what solvents they are using in terms of synthesizing the API. Ask the questions about what solvents they're currently using or what they foresee using in the future, so that you can prepare a methodology for residual solvents that will compile a list of solvents, all the ones that you will intend to look for. Now, if you are sourcing your materials from other vendor, you need to develop a supplier survey. Find out what are those residual solvents that the suppliers are using for the active pharmaceutical ingredients. Also, don't forget the excipients are important also if you're making the final product, because it would be good to find out from the supplier and request what is called the excipient information package. This is more like an MSDS for those excipients that you use to make your final product. Considering confirming the results in your laboratory. If they say it has, possibility chloroform, then you need to confirm that in your lab, and even develop a quality assurance, quality control procedure so as to determine that if an ongoing testing is necessary whenever you purchase this excipients. So every time you make a change in terms of where you source your material, it is a good thing to keep in mind. You need to check for the residual solvent impurities. An optimized solvent method is something that is accurate, sensitive, and fast, and you only have to analyze the solvents used in your process again. And you need to develop a methodology to meet your company needs. Now, this slide describes to you the three different classes of residual solvents. Class 1 would be solvents that are to be avoided because they have high toxicity. The second class are those that are solvents to be limited. You can use them, but they need to have some limited amount in the product. And then the class 3 would be the solvents that have low toxicity. So this slide describes to you the class 1 solvents to be avoided, and you see that the concentration limits are in the low ppm. The concerns are listed in this table, having the carcinogenic and toxic effect to humans. This next table may be difficult to read. Again, these are solvents that you can find in the book USP in the General Chapter 467. These are the class 2 with limited a permissible daily exposure. And you can see also that the concentration limit is a bit higher. The third class, which is the solvent with low toxic potentials, are listed in this table. I just wanted to go briefly in terms of how do you analyze residual solvents, and the major instrumentation for residual solvents would be the gas chromatography, and to consider the three main components of the GC, which is the injector, detector, and the column. So uh, make sure that when you're using the 467 described in USP, you can modify this, but it may require revalidation again for those conditions that you are changing in a major way. The injector can either be a headspace, which I think the Central Instrumentation Facility has this instrument, the headspace sampler. And this is the one that is described in the General Chapter 467. It has the advantage of reducing matrix interference, but the disadvantage is that some compounds are not detected using this methodology, headspace. You can do a direct injection in your GC because it is sensitive and it detects all the GC and 467 solvents, but the disadvantage is it is subject to a more matrix contamination in the column. So injection can be split, non-split. So headspace is specified in the General Chapter 467 in USP because, again, like I mentioned, it reduces matrix interference. So most of the drug formulation products are not volatile. So it is a good instrument to use. The disadvantage is that some of the compounds are not detected. And some of these compounds that are not detected using headspace are listed here: formamide, 2-ethoxyethanol, the ethanol types, the glycol ones. And this chromatogram shows you why. Because the first two big peaks, which are the methylene chloride and the benzene, has partition coefficient values of less than 10. One is 5 and the other one is 3. The partition coefficient actually is a expression where the concentration of the residual solvent concentration in the solution phase divided by the concentration of the residual solvent in the gas phase. So notice that if the partition coefficient for the 1,4-dioxane is 1,618, you would see a very small signal when you do a GC analysis of it. So that would be the reason why a disadvantage of using a headspace analysis. You can optimize in terms of the partition coefficient. You can optimize the system by adding salts to minimize the variation in the partition coefficient. For example, you have different residual solvents in your sample, and this lists down some of the salts that you can use to minimize the variation. You can change the dilution solvent, for example. You can change the heating temperature, and also you can reduce the solvent volume so as to increase the concentration, because the idea here is you have a sample dissolved in a liquid and you're heating up the sample. It takes time for it to equilibrate, but the smaller volume you have, the more concentration you have, but the molar ratio would be the same in the headspace. So the headspace is the one that you actually inject in the GC. In terms of equilibration time, you need to determine the equilibration time of your sample experimentally. So it is the ideal time is determined experimentally, but most of the solvents are ready after half an hour. So you would create this determination in your laboratory where you determine the response, monitoring the time. So again, it levels off after some time, and that is what is termed as the equilibration time. The General Chapter 467 as written can be optimized, again, so that a general protocol is intended to meet the needs of everyone. It has long analysis time, so you can modify it to shorten it. The sensitivity requirements are hard to meet for certain compounds. So again, it allows you to develop an alternative procedure. And at times, like I mentioned earlier, it does not detect all class 2 solvents because of the partition coefficient issue. It is not capable, it's not applicable in terms of some of the drug formulation. In terms of the GC detector that you are using, the ones that are typically used for residual solvents, as described in the General Chapter 467, would be the FID, flame ionization detector. Shown here, it has a wider range, but a good one to use would be a mass selective detector. And I believe Central Instrumentation Facility have this detector also, the mass selective detector. So a mass selective detector for GC has several advantages. It has high specificity, and it is good in terms of whether you are using the scan mode. It provides you at low ppm and even parts per billion in the sim mode. And also, the peaks are identified because of their masses. So it helps you in the identification, helps identify other unknown impurity peaks in your sample. So these are the advantages of having a mass selective detector. But you would need an internal standard so that you will have a high precision analysis. The disadvantages are several. It's an expensive instrument. It requires maintenance, so high maintenance costs. Also, the training in terms of who's using this instrument requires time and some effort. Also, it's difficult to match the accuracy and precision obtained from other detectors such as the FID unless you use an internal standard. In terms of the column that you are using for alternative procedure for residual solvent, consider in terms of the inside diameter of your GC column. If you are trying to have a high resolution, you can use the 100 micrometer inside diameter GC column. It will provide you with good resolution. However, in terms of capacity, it's not going to be as much capacity, meaning if you have a lot of the residual solvents, the one that will provide you with better capacity in terms of the number of residual solvents you're using would be a wider diameter GC column, so something like 530 micrometer. So it has good capacity, but in terms of resolution, because the wider the diameter of the GC column, the lower the plate number. So the resolution is not as good as the smaller diameter GC column. In terms of the length of the column, as a starting point, the suggestion is to use something like a medium length GC column, somewhere in the 30 meters, because you can use a long GC column providing you with high resolution, but it's going to take you a while because it's going to be a long analysis. The opposite would be using a short column that has moderate resolution, but it's faster. So this would be the recommendation in terms of developing alternative procedure for residual solvents. So optimize your GC method so as you can save amount of time, and that many changes could be considered as an adjustment to the 467 general chapter, but some may require revalidation, and revalidation time is significantly reduced if your method runtime is also reduced. The benefit of an optimized method may outweigh the extra validation work. So now we go to the last alternative procedure development that is also gaining a lot of attention in many of the pharmaceutical industry, and this is the elemental impurities. Now, the old chapter for elemental impurities described in USP is a chemical reaction, and this has been replaced by two instrumentation. One would be, the procedure one would be the ICP OES, optical emission spectroscopy, and then the other one would be the ICP mass spec. And I believe, again, the Central Instrumentation Facility have this ICP-MS as an instrument for elemental impurities procedure. This provides these two methods that are instrumental, provides greater clarity. especially for those who are trying to develop an alternative procedure. So actually the 233 General Chapter actually has an actual procedure for elemental impurities, either for ICP OES or ICP mass spec. But if the specified compendial procedure listed in General Chapter 233 do not meet the needs of your specific application, you can develop an alternative procedure. The alternative procedure, however, must be validated and make sure that it is so suitable for your own use. So alternative procedure either can be limit procedure. So limit procedures are those that gives you yes or no results. And the quantitative procedures are the ones that gives you an actual number in terms of concentration. But there are certain requirements even for limit procedures. So for limit procedures versus the quantitative procedure, limit procedure must be demonstrated experimentally using an appropriate system suitability and reference material. So when you're developing a limit procedure, you know exactly what are the concentration, even if the final approach is a yes or no answer. So there must be known concentration to the yes, and there must be an appropriate acceptance limit of concentration. Also, the material or the mixture under test must be spiked. So spiking allows you to be specific in terms of your elemental impurities. For quantitative procedure, it would be the same, but the limit procedure must be done with repeatability, 6 independent samples, and acceptance criteria RSD is not more than 20%. In terms of specificity for limit procedure must be able to unequivocally assess each target element. So there's specificity even for limit procedure. For quantitative procedure, however, you need to establish accuracy. And accuracy would be a spike and recovery experiment. Somewhere between 70 to 150 would be acceptable for mean of 3 replicate preparation for each of the concentration. In terms of precision, when you're developing a quantitative procedure for elemental impurities, the repeatability can either be using 6 independent samples of material under test, and then the acceptable criteria is an RSD of not more than 20%. You need to do intermediate precision also, which is a different day, different instrumentation, different analysts, and an N or a number of analysis for 12 would require an RSD of not more than 25% for each of the target element. So quantitative procedure also requires specificity. And this specificity is must be able to unequivocally assess each target element in the presence of other components. So you need specificity. In terms of limit of quantitation, range, and linearity, it must be demonstrated by meeting the accuracy requirements for the quantitative procedure. That's all I have for the development of alternative procedures, starting with the identification test, assay, organic impurities, residual solvents, and elemental impurities. Thank you for your attention. Chris?
Thank you, Doc Lonel. We'll answer your questions later after the discussion of the third topic.
I think we're on target on time, right, Chris?
Yes, sir.
Okay.
We are.
Good. Thank you. So this third presentation is probably the one that is the basic in terms of method validation, verification, and transfer of analytical procedures. And I understand the audience varies from high school to college graduates, or even staff, or from industry, regulatory. So this is more of the most basic of all the three presentations. Again, before I make any presentation or presenting this topic, the disclaimer is that this presentation are my views and not the views or policies of the USP, FDA, or ICH. In terms of outline of my presentation, third one is simple because it will first describe the validation of analytical procedures, then verification of analytical procedures, and finally the transfer of analytical procedures. These are the things that I typically teach in pharmacy schools and graduate schools. So the general chapter validation, verification, and transfer. Again, this is a review. Validation is required when an analytical procedure is used to test a non-official article, or an official article is tested using an alternative procedure. So this is covering the general notices 6.3. General chapter 1.2 to 6, which is the verification of compendial procedure. When is this required? This is required when the first time an official article is tested using a USP procedure. And finally, the ones that is general chapter 1.2 to 4 would be the transfer of analytical procedure. And when do you use this? You'd use this when you are moving a non-compendial procedure, alternative or internal, moving it from one laboratory to another. So let's start with the validation of analytical procedure. So in analytical method validation is actually performing within the acceptable standards of reliability, accuracy, and precision for its intended purpose. So validation is the act of confirming that the method does what it intends to do. So also the validation guidelines in general chapters 1.2 to 5 is a guideline, but as indicated here, validation activities is open to interpretation and practices differ greatly between organizations. Meaning, if there is a validation procedure from one organization, say one pharmaceutical lab A, they may be performing precision like for 12 samples, and for another laboratory, laboratory B, they may be performing precision using 6 samples, 12, 8 samples. So the details in terms of actual validation protocol varies from one organization unit to another. But the goals of validating analytical procedure are several-fold. Analytical procedures are of equal importance, meaning whether you are developing and validating a method for identification or for assay or for organic impurities, all of these have equal importance because they are intended to perform a specific function. And then in general, these validated analytical procedures are used in different ways for in-process testing, release, acceptance, even for stability testing. But again, you would want to have a validated method that minimize variation in terms of results. Again, the recurring theme for validation is it is suitable for its intended use. So the method is a planned systematic collection by the organization or by the applicant in terms of supporting their procedure. So this is, again, mentioned in the general chapter 1225. For those who are using the USP book, which is the monograph, there is no need to do validation simply because the procedures that are described in the USP book have been validated, and you can use them in another laboratory without doing any method transfer or transfer of analytical procedure. Definition of validation based on the United States FDA is consistent with USP and ICH. This is based on their document guidance for industry, which is focused on method validation, focus on the suitability for their intended use. ICH definition. Again, it's consistent. It is suitable for its intended purpose. But I wanted to clarify a terminology called the specification. So specification is referring to the analytical procedure and acceptance criteria. Many people refer specification as the acceptance criteria only. It has to include the analytical procedure and acceptance criteria together. So specification also is important to understand, because when we go to trying to understand verification, specifications are chosen to confirm the quality of a drug substance and drug product. It is not intended to describe the full characterization of the article, and it is focused only so that it ensures the safety and efficacy of the drug substance and the drug product. So that's the purpose for specification. Now, these are the lists of what is termed analytical performance characteristics: specificity, accuracy, precision. Some of you are familiar with these things, but let me just go quickly through it so that we have a common understanding, and we know what we're talking about. Specificity would be discriminating between the analytes of interest and the other components. What other components? So if you have a sample, for example, a drug product, there may be, when you put them in solution, there may be some impurities, degradation products, there may be some excipients in there that could interfere in your analysis. So specificity is important so that what you are trying to measure and what you actually see as a signal from your instrument is actually what you're trying to monitor. So the response observed is due to the single analyte of interest. Again, we are in the age where we have a more sophisticated instrumentation, and even at times, even optical isomers need to be isolated and separated because they have different um, biological activities. The degree of specificity testing varies depending on the method type and the stage of the validation, and that specificity should be evaluated continually through the drug development process. So I showed you the drug development process earlier in a graph, all the way from the development up to the product approval, even to post-manufacturing. So specificity needs to be continually evaluated, because at times there are impurities, degradation impurities that shows up, and they may interfere in terms of the specificity of your method. The accuracy is simple. This is an agreement between the accepted true value or a reference value against the actual results obtained. So either you know exactly what it contains, or you are using a reference material. And then accuracy, how do you do it in the validation? You actually do the accuracy by spike and recovery of samples. In terms of the definition of precision between ICH and USP, ICH have three things about precision. One would be the repeatability. And it's the same definition for USP, which is the same laboratory for a short period of time, same analyst, and same equipment. ICH also uses the term intermediate precision, which is within the laboratory, different days, different analysts with equipment on the same laboratory. And then the ones that are not typically done would be the reproducibility, which is what is termed interlaboratory. This would be, for example, analysis from a one laboratory, say for example at the FDA, or another analysis done in a academic, and then the third one would be something from an industry. So those are three different labs. Reproducibility. Make sure that we understand the term robustness and ruggedness, because robustness, again, is a measure of performance of a method when you are changing or deliberately changing, making a small change in terms of the method condition. These method conditions that are being changed are the ones that are identified to be critical. Ruggedness, however, is a precision term. It is similar to reproducibility. These are different labs, and different instrument, and they are not being widely used because it results in confusion at times. So definition of linearity between ICH and USP is the same. These are the results with which directly proportional to the concentration. However, USP has a little bit of a terminology because these are results that are directly or by a well-defined mathematical transformation. So at times, you may need to change some of the or transform some of the signals in, for example, or the concentration in terms of logarithmic. There are semi-log linearity curves. So it's not necessarily only signal versus concentrations. At times you need to do a semi-log. So that is the definition of linearity. Semi-log versus signal is applied, for example, if you're using an ion selective electrodes. Definition of linear range. If you have completed the linearity, you would want to uh, know the interval which between the upper and lower concentration is, and this is termed as range. In terms of limit of detection, same consistent definition between ICH and USP, which is this would be the lowest amount of analyte in a sample which can be detected. So this is not necessarily within the linear range. So, but it would be a signal that you can detect. At times, the limit of detection is close to the signal to noise ratio, somewhere between 2, 3, or even higher. Limit of quantitation, however, is different because limit of quantitation, as most of you know, would be the lowest amount of analyte in a sample that can be quantitated. So limit of quantitation would be, if you have a linear range, that would be the lowest concentration or the low range. So the limit of quantitation means that you are still in the range where the signal is proportional to the concentration. Data elements required for the validation is described in this slide. Now, in USP, it can be categorized into 4 different categories. The first category would be focused on the major components, and these are termed the assay. Category 2 would be impurity tests. So impurities can either be organic, inorganic, residual solvents, elemental analysis. So category 2 are focused on impurities in drug substance and drug products. Category 3 for data elements required would be what is termed the performance test. This is applicable to drug products. These are tests such as the dissolution test, disintegration, drug release, particle size. And then the 4th category is the term identification test. So it identifies the active ingredients. This slide describes to you the data elements required for the validation if you're doing a validation based on USP General Chapter 1225. Again, it depends on the analytical performance characteristics, and also it is dependent on the category type. As you can see, for example, if you are validating a category 1 assay, everything here listed is needed except the detection limit and quantitation limit. And so you would apply the same way to read this table. So as in quantitative impurity test, validation is required for everything except detection limit, but you need quantitation limit. It is not applicable for category 3 performance because performance is only applied to drug products, not drug substance. But here, the precision is applicable for drug products. Category 5, which is identification, everything is not needed except specificity. So characteristics are the same that are described by the ICH and USP. All these 7 validation characteristics are consistent, except in ICH, their category for identification and the required analytical performance characteristics is the same as that of USP. Impurities, again, the same for both the quantitative and limit tests, except for the assay. Their assay includes also performance tests such as the dissolution, content uniformity, and potency. So they bunch together the assay and the performance tests versus USP separates them in the previous table that I have shown you. So that would be a difference between ICH and USP. Critical step, first step in the validation. It is important that you need to know the purpose of the method. So you need to clearly state that. And per ICHQ2, the objective of the analytical procedure must be clearly understood. So there must be a statement of the validation conditions. Uh, so typically, when you do validation, it is assumed that you have already performed method development. In years ago, method development is something that doesn't need to be documented. It has become important that method development is a requirement, and you need to document all your method development before you do all the validation. It is good to document all those method development because when you have difficulty with your methodology in analyzing your sample and you have to go back and analyze the conditions, what you have to change. For example, you're trying to do an alternative procedure. It is good to have a background information in how you develop the method. If the conditions are found to be mitigating against using the method, they should be indicated, meaning specific instructions such as analyze the sample within 2 hours, or you cannot go beyond plus or minus zero point 0.2 in terms of pH. Those are the specific items that needs to be included in the validation. The stability indicating method is something that the United States FDA has required for many of the pharmaceutical industry. Stability indicating methods are, as a term, it's able to monitor the stability of the drug product and drug substances. Now, for many years, the analytical method of choice is, for example, TLC or titration, and these are not stability indicating procedure. For example, if it's an acid-base titration, the base will titrate any acid regardless of what type of acid it is, as long as it has the acid functionality. While in HPLC, that's the reason why it's being used so much widely for many years, the HPLC allows you to do separation of the components before the quantitation. To demonstrate the specificity of a stability indicating procedure, all known interferences must be tested, or you can even develop a protocol to have a laboratory stress condition, meaning you stress the sample by a different condition, adding peroxide, adding acid base, adding light, and so on and so forth. So you can accelerate the temperature and humidity condition of your sample. This is to force your sample to degrade so that you can monitor the degradants while still maintaining specificity. So the validation data should indicate robustness evaluation. As I have mentioned, robustness is something that is done mainly at the method development stage. Nowadays, it has been mentioned in the USP General Chapter 1225, which is the validation. Also, when you're submitting your dossier to the FDA or USP, robustness data is now required. So we go to the verification of analytical procedure. Verification is actually not the same as validation because in the verification, validation is challenging the analytical method using a well-defined sample, while verification challenges the analytical environment using a well-defined method. So the applicability would be for drug substance, drug products, and excipients. The application can be on different analytical methodologies, titration, chromatographic, spectroscopic, and the definition of the verification, it is the confirmation by examination and provision of an objective evidence that the specified requirements has been fulfilled. okay, and requirements. This is based on the Code of Federal Regulation. It is not actually the same as validation because only selected analytical performance characteristics are chosen so as to generate the appropriate relevant data. It is not a repetition of validation. So verification is needed so as to make sure that the laboratory performing the test can reliably perform with precision and accuracy the method as it is intended. So verification, uh, there are no general protocol for verification. This is one thing that whenever I teach this in other Asian countries, there's a lot of misunderstanding in terms of verification. But the guidance in the general chapter 1, 2 to 6 verification is only a guidance and it's not specific. I will, however, provide you with a recommendation on how to do verification, and this would be more or less targeted by how the US FDA expects the pharmaceutical company to do verification. Based on the GMP, it says that the suitability of all testing methods used shall be verified under actual condition of use. Also, verification is done only at the beginning, the first time. It is not done on a routine basis because once a method is verified that it is capable of analyzing the samples, then that would be it. Verification requirements should be based on assessment in terms of the complexity of the procedure and material. Now, when we were discussing about the validation, which is I mentioned about internal factors, which is focused on the analytical performance characteristics. The General Chapter 1, 2 to 0, which is a holistic approach in terms of the validation, verification, and transfer, looks at everything. What is everything? And the environment that is referred to in the verification is focused on procedure related. What are those procedure related? Those are dependent on the analyst level of training, laboratory that is being used, the type of procedure associated to the equipment. Second thing would be it is material related. Sample, article to be tested, the reagents, the matrix. So these two uh, external factors are considered in the verification process. If this is the table that describes to you the data elements for validation, this is the one that I was going to provide you as a recommendation for to perform verification. But this first slide would be for drug substances and excipients. So it would depend on the analytical method that is being used. It is also dependent on the category of tests, whether it's category 1, assay, 2, 3, and 4. So for example, if you are doing a verification of the product using the assay method and your analytical method is HPLC, you only need to do precision. That's all you need to do. For category 2, for impurities, for a quantitative method using a using an HPLC method, you have to do precision, specificity, and limit of quantitation. Same thing with the category 4 identification, but category 3 performance for drug substance and drug excipients is not applicable. However, this next slide, which is for drug products, now you have a category 3 performance verification. If you are using an HPLC, then you need to do precision. It would read the same way. So it would depend on, again, analytical method and the category of test that you are trying to verify. In terms of how does the industry define in terms of what do they do when we say precision, when we say accuracy? When they say accuracy in a verification mode, it is they determine the recovery at the specification range, meaning they need to know what is the specification range. I mentioned to you what is the definition of specification range, right? Earlier. Precision would be determine the repeatability only in the specification range, Specificity is a test for peak purity and matrix effect. Limit of detection is verification of detection at 50% of the specification. And finally, the limit of quantitation would be the verification of quantitation at 50% of the specification. Example of a verification of drug substance and drug products. And I will go through this one because this is something easier to see rather than the summarized two tables that I've shown you. For example, if you are doing a verification of a drug substance, such as the sodium chloride, your method is titrimetry. So precision may be needed. So that would be something that you only need to do. However, if you are doing a verification for a drug substance using an assay and your method is HPLC, then you need both precision and specificity. Now, if we go to the other categories for drug substances such as impurities, then you need to do precision, specificity, and limit of quantitation. Category 4, identification test would be for a method that is IR, UV, or HPLC, then specificity is needed. If we go into the drug products for a category 1 assay using an HPLC method, then you need those three things: accuracy, precision, specificity. Category 2 for impurities using an HPLC method would require precision, specificity, and maybe limit of quantitation. For performance methods such as dissolution, you need to do precision. And finally, for category 4 identification tests, again, focus on the specificity. So to summarize verification, the intent of this chapter is to provide general information to laboratories on the verification of compendial procedure that is coming from USP that's being performed for the first time so that as to yield the acceptable results. It is not intended for retroactive, meaning if you have done it for the first time, you don't have to keep on doing it. And also verification consists of only selecting the analytical performance characteristics as opposed to doing everything in general in the validation. Finally, the last part would be the transfer of analytical procedure or method transfer. This would be the general chapter 1, 2 to 4, transfer of analytical procedure, which is the last general chapter that was added in USP. And this is, again, the transferring unit is moving the method to a receiving unit so as to ensure that the receiving unit has the procedural knowledge and ability to perform the transferred analytical procedure as intended. And this is not applicable to USP monograph because you can use USP monograph at any time in any other lab without doing any formal transfer. So when is transfer needed? When, for example, you are moving a procedure or method from R&D to QC lab, site A to site B, QC to contract lab, instrument A, you're changing to instrument B. And then again, it is a risk-based approach because it depends on external factors, experience and knowledge of the analyst, specification of the product, the complexity of the analytical procedure. So you need to evaluate the extent and the type of analytical transfer procedure that you're going to implement. I mentioned there are 4 of them. So it all depends on whether they are validated or not. You've seen this slide earlier that a transfer waiver can be done, a revalidation, or a comparative testing. If it is not validated, you can do a co-validation with the transferring unit. Just to summarize, transfer waiver, what are those? Those are for new product composition that is comparable to that of an existing product. So there's really no change in the product. Also, the procedure transfer is the same, meaning that your staff moving from one site to another and the same equipment is being used. So a transfer waiver is possible to do that. Comparative testing, however, which is the most common transfer analytical procedure protocol, require the analysis of a predetermined number of samples from the same lot by both the transferring and receiving unit. Revalidation would be something repeating some or all of the validation experiments, but this would be option that has the most time consuming, because it's, again, the receiving laboratory is going to be performing the validation. The co-validation is a bit better because it is both a collaboration between the transferring unit doing some of the validation, and then the receiving, which will also do some of the validation. This is used when procedure is not fully validated. So I have listed some of the resources that some of you would want to pursue and find out more. And these are available online. And the last slide actually lists down all this relevant information. The FDA coming from the United States Food and Drug Administration, ICH, which is the international regulatory, provides international regulatory guidelines for drug substance and drug products. And also, if you have the book for USP, you can refer to some of this general chapter. Thank you very much for your attention. It's my honor to present all these 3 topics, and I hope you learned something from it.